the majority of the presentations everywhere, the first slides are on behalf of the institution, like Hadassah. You put it at the end. That's okay. It's not business-wise. Okay, thank you. Now we'll continue with Dr. Michal Israel. It's kind of continuation to what we've heard now, but this is the first presentation actually will deal with Kadima STEM, except for mentioning all the time Michelle, so this is the first presentation. Michal is the Vice President of Research and Development for ALS and neurodegenerative diseases at Kadima STEM. She has been with Kadima STEM since 2009. Prior to joining Kadima STEM, she performed her doctoral work in molecular genetics at the Weizmann Institute of Science in the Professor Revel Laboratory. Dr. Israel was a central part of the team that invented and developed the technology which became the basis for Kadima STEM AstroRx cell therapy product. Michal.
Okay, so today I'm going to tell you about the road that led the first clinical trial with AstroRx. So AstroRx are the human embryonic stem cells derived astrocytes that we developed in Kadima STEM. So the vision of Kadima STEM, which is under the supervision of Professor Revel, is actually that we are producing cells that actually are meant to replace, restore, or repair, and repair the functionality of disease and malfunctioning and organs in various degenerative diseases. And how do we do that? And our vision is actually transplantation of healthy and functional cells which can actually release patients from the burden of degenerative diseases. So we are actually producing spare parts. Okay? So how do we do that? So our platform technology is by the use of human embryonic stem cells or induced pluripotent stem cells, which are cells that can actually expand indefinitely, and they can give rise to any cell type of the human body. Okay, so we take these two features, we expand the cells, use them as our starting material, and then differentiate them towards two cell types. The first cell types are astrocytes, human astrocytes, and the second one are the insulin-producing cells, which Kfir will elaborate on that later on. So this is the main idea of the company. So the only time that I will use the word stem cells is only as the starting material, and we are actually producing functional, healthy cells that can actually replace the malfunctioning of specific diseases. So we are focusing on ALS and diabetes. So ALS is amyotrophic lateral sclerosis, also known as Lou Gehrig disease. Around 450,000 people are affected worldwide. And in this disease, the muscle cells that actually control our movement are dying due to the death of motor neurons. And around 90 to 95% of the disease are familiar with genetic background, and 5 to 10% of diseases are, vice versa, okay? So 90 to 95% of the disease is sporadic with unknown genetic reason, and 5 to 10% of the disease is genetic with mutations such as the human SOD1. We will encounter that later on as the animal ALS animal model, the human SOD1. There are several other mutations such as the TDP, C9orf72, and etc. And since disease onset, which usually takes place around the age of 50 to 60, but there are cases where the disease appears much earlier, the survival rate is between 3 to 5 years from disease onset. And to date, there is no real effective treatment for ALS, except from 2 FDA-approved drugs with very moderate effectiveness. One of them is the Rilutech, and the other one is called the Radicava. And the main rationale behind using human astrocytes is that in ALS disease, the supporting cells, the astrocytes, were found to be malfunctioning, and they failed to actually support the dying motor neurons. So they fail to actually produce neurotrophic factors that can induce the survival of motor neurons. They fail to protect from oxidative stress. They fail to uptake toxic factors such as glutamate, for example, which is a neurotransmitter which once exists in the synaptic gap for too long time actually can cause death of motor neurons. So the main idea is actually take healthy astrocytes, transplant them, and replace the malfunctioning of the endogenous human astrocytes. Okay, so very simple idea. Let's see how we do that. Okay, so in CadimaStem, we actually start with the human embryonic stem cells or induced pluripotent stem cells as our starting material, and we mimic the embryonic development by using all kinds of cell suspensions, factors, and actually mimicking the embryonic development. And we have defined a very nice protocol where we start with these cells. We differentiate them, and we obtain the progenitor cells, the astrocyte progenitor cells that can be kept frozen as cell banks, so we don't need to repeat this process all over again. So at the moment, we have clinically grade APC, astrocyte progenitor cell banks, for our future use. These cells are kept frozen, then they are thawed, expanded a little bit, differentiated for a very short period of time in order to have a committed astrocytes, and then the idea is actually to transplant these cells intrathecally, as Professor Tamir Ben-Hur mentioned, into the cerebrospinal fluid, to the CSF, with the idea that the cells will distribute throughout the neural axis. They will reach the upper motor neurons in the brain, the lower motor neurons along the spinal cord, and then they will have the effect, the therapeutic effect. So in order to do so, it's a nice idea. So in order to do so, we actually need to do the process, and we need to characterize the cells. We need to follow all the regulatory demands of the FDA, of the Israeli Ministry of Health, and etc. So this is how it looks like. We start with a nice xeno-free clinical grade starting material of human embryonic stem cells. We have a differentiation process where we have the neural spheres, which are attached into a very specific matrix. These cells are then harvested, expanded as single cells, and then fully committed to our cell product with very, very high purity of cells of human astrocytes. Okay, so as we said, okay, we need to actually follow the FDA and the Israeli Ministry of Health demands, and we need to fully characterize the cells in terms of their identity, their purity, and actually have potency assays to actually determine their functionality. So we differentiated the cells, and we have a very nice panel of human-specific astrocyte markers, which we are obligated in terms of acceptance criteria. So as you can see here, we have the GLAS, the CD44, the GFAP, the aquaporin as astrocytic markers. So we have a very, very nice purity of these cells. And in addition, we need to prove that actually the starting material, the pluripotent stem cells, which are very, very risky because they can give rise to teratomas and all kinds of cell masses, are not actually present in our culture. So we validated this system, and we have a very nice panel of pluripotent stem cells marker like TRA160, SSCA4, EPCAM, etc. And we can actually show that there are no pluripotent stem cells markers in the culture. So next, okay, so we identified the cells, we actually measured their purity, and now we want to see whether they are functional in vitro. So for that aim, we did several potency assays. So one of them is the glutamate uptake. As we mentioned before, we put a very specific amount of glutamate in the media, and we want to see whether these glutamate levels are disappearing in the media with the presence of human astrocytes. So you can see here that in the presence of AstroRx, or the human astrocytes, the glutamate actually disappears from the media. The cells actually uptake glutamate from the media, and it's very similar in comparison to spinal cord astrocytes that we used as a control. And in terms of other cell types, fibroblasts don't do this activity, so we had it as a negative control. So next, we wanted to see whether the cell can actually secrete neurotrophic factors that can actually promote the survival of motor neurons. So first, we looked at the classical ones, the BDNF, the VGF, the GDNF, and we saw that these cells can actually manufacture, in the left bars, these factors, and these factors are secreted to the media. And then we asked, okay, are there additional factors which are secreted to the media? So for that aim, we did a secretome analysis of around thousands of factors which are tested. And we can actually see that a lot of well-known neurotrophic factors are actually secreted by these cells, and there was a very nice correlation with ALS for some of the factors such as TIMP1, TIMP2, OPN, which are reduced in the disease, and using our cells, when actually our cells secrete these factors that can actually promote the survival of motor neurons. And we wanted to see the cross talk between the astrocytes and the neurons. So we did a co-culture between our human astrocytes and the neurons themselves. And we saw that once we grow the astrocytes with neurons, it actually promoted the survival of neurons and accelerated their neurite outgrowth and axon outgrowth, indicating their therapeutic effect in vitro. So next, we had to move to in vivo studies to see whether the cells can actually distribute throughout the CNS once we transplant them intrathecally. Do they survive along this time? Do they maintain their astrocytic identity? Do they have an effect on the progression of ALS disease? And finally, and very importantly, are these cells safe? So for that aim, we used the human SOD1, the genetic type ALS model, the high copy number, which is a very severe animal model. From onset, between 2 to 3 weeks, the animals actually die. And we measured several factors, such as the survival, the grip strength, the rotor road, which are motor performance measurements. We also measured the muscle weight loss, or the body mass. Once you lose your muscles, the body mass actually reduces. And the neurological score for the disease. The cells were transplanted intrathecally, very similar to the route of administrations that we are using for our clinical trial. And these are the results. So as you can see here, once the cells were transplanted, there was a very significant delay in disease onset. There was a prolongation in the survival rate of the animals. And in terms of motor performance, we actually maintained their motor performance for a longer time once we measured it with the grip strength and once we measured it with the rotor rotation, indicating their effectiveness for this kind of measurements. And it is very important to say that these studies were made in the rat model, but we also did it with the mouse model, actually transplanting the cells intrathecally through the cisterna magna, and we had very similar results. So we have two types of models that we had an effect of the cells. This is in terms of body mass. Okay, so you can see that once the cells were treated with AstroRx, they maintain their body mass for a longer time. And this is a short movie. Let's see if it works. Okay, so you can see on the left, this is the rotor road. These are the transplanted animals. On the left, you can see a sham-injected animal. On the right, you can see an AstroRx-injected animal. And you can see that the AstroRx-injected animal maintained for a longer time on the rotor road and maintained their motor performance for a longer time. So what about cell distribution? So in order to actually assess or answer this question, we did an intrathecal injection of the AstroRx cells into immune-deficient mice. Here you can see the brain, you can see the spinal cord. We transplanted the cells through the cisterna magna intrathecally, and we monitored for 4, 17, and 9 months after transplantation whether the cells survived, do they maintain their cell mass, do they actually proliferate, where are they located at. And as you can see here, okay, once we transplanted the animals here, you can see different levels of the brain, okay, and you can see along the spinal cord, the cells are here labeled with DII, okay, which is a fluorescent dye, and you can see that the cells actually attach to the pia mater, to the meninges, and extract their therapeutic effect to the surrounding CNS parenchyma. So you can see that actually the cells are enveloping the CNS. And in terms of cell survival, we saw that after 9 months, the cells actually survived, and the cell mass or the number of cells maintained very, very similar. So there was no extraordinary proliferation, and using Ki67, which is a proliferative marker, we saw there is no proliferation of these cells. So the next question was whether they maintain their identity. So for that aim, we actually stained with a GFAP, which is a human astrocyte markers. These are the images of the cells, how they look in vitro. So this is GFAP staining S100 beta, which is another astrocytic marker. And you can see here that once the cells are injected intrathecally, they are GFAP positive, they are astrocytes, they maintain their astrocytic identity. And this is a closer look, and you can see that actually it envelopes the nerve bundles, which are extracted to innervate different muscls in the body. And it was very nice to see that actually there was no formation of tumors or neuroteratomas after 9 months after injections, indicating that the cells are actually safe. And this was, again, under the roof or under the umbrella of the FDA guidelines. Okay. So also in terms of biodistribution, we wanted to know if the cells actually maintain in the CNS and they do not distribute to other cell organ or to other organs in the body. So for that aim, we used the PCR, very sensitive method, and we saw that they're maintained in the CNS and they do not distribute to other organs. So after doing all this work, we manufactured the cells. We had a phase where we transitioned from R&D to GMP. We have in Kadima Stem a GMP facility. We changed all our materials to be GMP suitable. So we have all the chemicals, manufacturing, and controls. We have the preclinical studies indicating that the cells, that we know the identity of the cells, we know the purity, we know how the possible mechanism of actions. And in addition, we showed that the cells have an effect in the animal model and that the cells are safe. Building all this regulatory package, we submitted to the Israeli Ministry of Health here in Israel, and we were approved in March 2018, like 4 months ago. We were approved with a phase 1/2A clinical study at Hadassah Medical Center at the neurology department. The principal investigator is Dr. Mart Godkin, which is an ALS expert here in Israel. It is also a sub-investigator, and we are doing it in the HCRC, which is the it's actually the clinical trial center at Hadassah, so everything is well supervised. We have an external CRO, which actually supervises all this clinical trial, which is Cato. It's an international company. And the main objective of the study is actually a phase 1/2A open-label dose-escalating clinical study to actually evaluate the safety. This is our primary endpoint, the tolerability and therapeutic effects of transplantation of AstroRx cells. Okay, so this is the main idea. And we have 4 experimental cohorts. We start with 5 patients, which are going to be injected with 100 million cells. The next dose are 250 million cells, which are going to be injected intrathecally to these patients. The third cohort is 2 injections of 250, the mid dose. And the final cohort is 500 million cells. So as we said, the first endpoint is safety. The second endpoint is actually measuring some kind of an efficacy, where we have 4 types of measurements. One of them is the ALSFRS. The second one is the handheld dynamometer, which we actually measure several sets of muscles. The grip strength, the SVZ, which is the slow vital capacity, and etc. And we hope that by the end of the study we will have results of the safety and, of course, some smell or maybe very strong efficacy data. So it's very important to say that in many neurodegenerative diseases such as Alzheimer, multiple sclerosis, etc., astrocytes have a pivotal role in maintaining the well-being of motor neurons or neurons in general, and also on oligodendrocytes. So we can use this technology as a platform in order to actually inject the cells in other neurodegenerative diseases and actually see whether we have a therapeutic effect in these diseases. So this is our daily life, how it looks at CadimaStem. We have now a very strong manufacturing activity. These are some of the people which are working in the lab. We have also an R&D activity. And this is actually the formulated syringe which is going to be used for our phase 1/2 clinical trial. So in this opportunity, I would like to thank Michelle and tell you Mazal Tov on behalf of all CadimaStem, and that's it.